Good evening, ladies and gentlemen, and welcome to this year's Ian Beveridge Memorial Lecture. My name is Fraser Allen, and I'm the Head and Dean of the Sydney School of Veterinary Science. Before we begin proceedings, I would like to acknowledge the traditional owners of the land on which we meet, the Gadigal people of the Eora Nation. It is upon their ancestral lands that the University of Sydney is built, and I would like to pay my respects to their elders past, present and emerging. As we're here today for the Ian Beveridge Memorial Lecture, which focuses on the interaction between humans, animals and our natural environment, it is poignant to note that the Gadigal people maintained perfect harmony between humans, plants, animals and the environment for tens of thousands of years. We have a lot to learn from them. Tonight's lecture is held in honour of Emeritus Professor William Ian Beveridge. He was a veterinary alumnus of the University of Sydney, graduating in 1931. His working life was devoted to fighting disease. He began his illustrious career working on sheep at the CSIR McMaster Laboratory before moving to the USA to undertake a fellowship. During his career, he worked at leading international institutions such as the Pasteur Institute and the University of Cambridge. Ian understood the importance of One Health, the interconnectedness between human disease, animal disease and the environment, and devoted much of his later career to the World Health Organization, controlling diseases that affect both humans and animals. It now gives me great pleasure to introduce tonight's speaker, Professor Ruth Zadoks. The School of Veterinary Science is delighted that Ruth has recently taken up a role with us as a Professor of Production Animal Health. She is a veterinarian whose research focuses on promoting the health, welfare and productivity of food-producing animals and the people that look after them and those who use those products. Her main research interest is bacterial infectious diseases of livestock, fishes and people. As a molecular epidemiologist, she uses DNA-based methods to refine the characterization of disease-causing organisms. Such DNA fingerprinting or strain typing provides detailed insights into the sources of disease-causing agents, how they are transmitted, and how diseases manifest. By understanding these mechanisms, she can provide advice to farmers, veterinarians and policymakers about ways to improve animal and human health and productivity. We will learn more about her work on Group B Strep tonight, but I can tell you that it spans 6 continents, a range of host species, and includes the development of strategies and tools to reduce disease risk in people and animals. Ladies and gentlemen, please welcome to the stage Professor Ruth Zadoks.
Thank you very much, Professor Ellen Fraser, for that kind introduction. Good evening, everybody. Welcome, and thank you for being here tonight. There are a few people whom I know can't be here tonight because their houses and families are under immediate threat of the bushfires. And I'm sorry for that. I hope they'll be safe. I hope that all of us, particularly at the Camden campus, will be safe tomorrow. And actually, I hope that all of us will be safe throughout the fire season. But while we're here, I'll try to entertain you with stories about Group B Strep in babies, adults, and animals, and answer the question, is there a link? Now, before I start answering that question, I would actually like to ask you a question. Has anyone seen Julia's Home Delivery with Bill Bryson the week before last? Home Delivery, yeah. Me and a few other people, maybe a few more people, but just shy. So, Bill Bryson is an author I like. He's written a lot about countries where I've lived, the US, the UK, Down Under. It's one of the things I read before deciding whether I would move here, take the risk with the spiders and all the rest of it. And Bill Bryson said that he writes for pay. Now, that in itself is not a sin. You could argue that to some extent, as scientists, we write for pay. That's fine. But his most recent books have been about science, not about travel, but about science. And the reason Bill Bryson writes about science and gets paid to do that is because scientists do not know how to communicate with normal people. You're normal people. Well, some of you anyway. I am a scientist. Bear with me. Right. The other thing I'd like to say is that, as Professor Allen explained, this is the Ian Beveridge Memorial Lecture, who was one of the leading lights really in One Medicine and One Health. And different people have different interpretations of what exactly that means, and then there's global health and planetary health and various permutations of the concept, but it all boils down to one thing, and that is that human health, animal health, and environmental health are linked. We're all in it together. And to bring people together, there's nothing like having a common enemy. So let me introduce you to tonight's common enemy as I try to answer the question, Group B strep in babies, adults, and animals, is there a link? And this is our enemy. Rather beautiful, I think. It's also rather deadly. And, as some of you are aware, but maybe not all of us, it's present here in this auditorium. On average, 20% of people carry Group B strep. 20% of people, it means 1 in 5. Which means that between you, the person in front of you, the person behind you, person to your left, yeah, have a good look, person to your right, one of you carries Group B strep. Statistically speaking. It wouldn't actually be you, would it? Where do we carry Group B strep? So carrying means that we have it with us in our bodies without it causing disease. So this is from a website from the Australian government, from the Department of Health. And this reminds me of something else I should say, and that is that I'm a veterinarian by training. I am licensed to work as a veterinarian and to prescribe medication and treatment to animals in the Netherlands. I'm not licensed to do that here in Australia. I'm definitely not licensed to give medical advice. So if anyone has any concerns about human Group B strep, any questions about diagnosis, prevention, or treatment, please see a healthcare professional. I am a scientist and a veterinarian, not a medically trained professional, so I can't answer questions about your personal health. So Group B strep, according to the Australian government, is a common bacterium that's generally found in our gastrointestinal tract and the urethra, which essentially means that if we don't wash our hands after going to the bathroom, we carry Group B strep on our skin as well. And this website specifically mentions the vagina, which is part of the human body, and that's the truth and nothing but the truth, but it's not the whole truth. Men are equally as likely to carry Group B strep in their urogenital tract or the gastrointestinal tract. Some studies say that the carriage rate is a bit higher in women, some studies say that the carriage rate is a bit higher in men. There are behaviors and activities that are linked to that carriage rate, but all of us can carry Group B strep. Now, that in itself isn't a big problem, apart from the fact that if a woman is pregnant, then Group B strep carriage may lead to Group B strep infection with consequences for both mother and baby. And that's where the deadly part of Group B strep comes in. This is a snapshot from part of a website in the UK from the Group B strep support group, and the name of that organization probably tells you enough. It's called a support group for a reason. It was set up by bereaved parents that lost a baby to Group B strep. And if you look at the second column from the left, at the bottom, also on the screen there, coping with loss. That's what Group B strep does, unfortunately, to babies, can do to babies, and even to their mothers. Some babies recover completely fine. Some babies develop permanent neurological symptoms, which means that their impaired in their cognitive and physical abilities, some babies do not survive at all. That's why we need to worry about Group B strep in babies, and that's why in a lot of scientific literature and policy material and guidelines, you'll see that focus on Group B strep carriage by women. There's hope for babies, though, and their mothers and fathers, indeed. This is the conference website of a conference that was held last year in Cape Town, and that was the first international symposium on Group B Streptococcus. And a large part of that symposium was dedicated to discussions and presentations around a vaccine that's under development. In fact, there are several companies developing vaccines, and at least one of these vaccines is already in a safety trial. And hopefully that vaccine is a vaccine that we can administer to pregnant women to protect both them and their babies from Group B strep disease. This I want to be very clear about. This part of the question I can answer right here and now, and very simply, Group B strep in babies and animals, is there a link? No. For all practical intents and purposes, there is no link. Babies may develop early onset disease in the first week of life, or late onset disease after that first week of life. But in the Western societies where Group B strep is a concern in neonates, babies of that age don't have contact with animals, and by and large, they are not exposed to food of animal origin. So there's no link between Group B strep in babies and in animals. In the scientific literature, when you look at the evolution of Group B strep and where it came from and where it's going, there is controversy around this topic, but in everyday life, there is no link. What then about adults? This could be a very short talk. I know some of my colleagues are keen to have a cold drink, so let's make this short. Group B strep can be carried by us, but it can also cause disease in adults. So most of you here will know how to read the graph, but just to talk you through that quickly, the horizontal axis here is an age scale in decades, so 0 to 10 years, 10 to 20, 20 to 30, all the way up to 100. And on the vertical axis there you see a number, and that's just a number of bacterial isolates that were used in that particular study that you see cited at the bottom there. And in green, and I apologize to those people that may not be able to see green, but the tallest bars in the categories up to 30 and 40 are carriage isolates. So those were isolates that were collected from women that were screened during pregnancy to check their Group B strep status. All the other bars in this graph are not from pregnant women. They're actually from cases of disease, whereby the blue bars represent skin and soft tissue infection, with the youngest patient only being a year old, and he's in the 0 to 10 category, and the oldest patient in their 80s. The majority of infections in adults are urinary tract infections, shown in yellow, and that affects, again, all age groups, although maybe more so the elderly. That, until a few years ago, was how we knew Group B strep in adults, until something unprecedented happened in Singapore. And this gentleman is absolutely remarkable. I have not actually met him, but there's quite a bit of information on him online, and he's actually a public and motivational speaker. This gentleman, who you see here enjoying a meal with his family, the family is not in a photo, but there's a couple of photos online, and this is just one of them. He was one of several adults that came into hospitals in Singapore where normally they see maybe 1 or 2 cases of Group B strep a month. Now they saw 1 or 2 cases of Group B strep a day. And not just in one hospital, all hospitals across Singapore. So the medical microbiologists in the hospitals, the people that grow bacteria to see what makes patients sick and how we can treat them, got together and said, this is unheard of, what's going on here, and started an outbreak investigation. And in this outbreak investigation, they were helped somewhat by the characteristics of the patients, because most of them were middle-aged men that had been working and healthy until they got the strep disease, and of Chinese ethnicity, which is usually linked to people's behaviors, and particularly in Singapore, which has a very rich food culture, to the foods that people eat. And they interviewed the patients where possible, or their family members when the patients were too ill, and they discovered the cause of this outbreak and managed to stop it once they knew where it came from. This is another example of a victim of that outbreak, shown here with his wife. His wife has actually written a book about the experience. And although physically this man may look far more intact than the gentleman I showed in the previous image, this man lost his hearing, and he was a high-flying businessman making million-dollar deals, providing for his family. So in a way, the loss to this man, who essentially lost his identity, is almost bigger than that of the gentleman that lost his hands and his feet. Both of them lost their abilities thanks to a raw fish meal, and that's what the epidemiological investigation led to. All these people had eaten yusheng, which is a particular Chinese-style raw fish dish that's very popular in Singapore. And they checked not just the risk factor, but they actually went back and detected Group B Strep in the fish on the stalls. So then the authorities in Singapore issued a ruling that raw fish could no longer be sold from these stores. And you see here a typical food market in Singapore with on the left a stall that's still open, and on the right a fish porridge place that's been closed down. And what's interesting, I don't have a picture of it, but I know my colleagues in Singapore do. Before the authorities officially closed down the raw fish sales, on social media, messages were already circulating via WhatsApp mostly. My uncle had raw fish the other day. He's in hospital now. Don't eat raw fish. So social media, in this case, actually played a very positive role in limiting the outbreak. So for adults and animals, is there a link in case of Group B Strep? Yes, there is, definitely, because these people got sick from eating raw fish, and the fish carried the Group B Strep. But the answer is a bit more complicated than just, oh, there's a problem with fish. I think there's a problem with us, particularly with how many there are of us. So I want to take you back to population growth, which drives so many of the problems we're facing today. And again, there's a time scale on the horizontal axis starting in 2000 and ending in the year 2100. On the vertical axis, you see the total world population in billions. This is where we are now, almost 2020. I don't think we'll quite have the hindsight to understand all our problems. But when we look at a close-up of 2020, you can see that in the last few years, we've grown from a population of 6 billion to a population anywhere between 7 and 8 billion. And all these people need to eat. And the vast majority of these people will consume food of animal origin and animal protein in their diet. So in parallel with the population growth that you've just seen, there has been this growth in fish consumption. Again, there's a time scale running along the horizontal axis. There is fish consumption in millions of tons along the vertical axis, and this is data from the Food and Agriculture Organization of the United Nations. And there's a two-colored graph here. In orange, you see the consumption of wild-caught fish, fish that we catch from our rivers and lakes and seas. And I have friends in the Netherlands that think that anything to do with farming is the evil empire, and that will never eat fish that is farmed. They'll only eat wild-caught fish. And there are really two sides to that. On the one hand, they feel like they're doing something good. On the other hand, if everybody only ate wild-caught fish, there wouldn't be any fish left to catch. We need to manage the fish stocks such that they can replenish themselves before we outcompete the natural regeneration with our catching. And that's why we need aquaculture to meet the demand for fish and aquaculture products. So the bit in teal that you see on top of the orange part is the aquaculture derived fish consumption. And that line has continued to grow up and up and up. And if we extended that graph to 2020, then actually the aquaculture part would by now exceed the wild-caught part. So where does that consumption fish come from? A lot of it comes from China, about 70% globally, and of the rest, about 70%, give or take, comes from Southeast Asia. So let me take you on a short journey to Southeast Asia, to Vietnam in this case, where traditional Vietnamese life unfolds quietly in the background, and we are in the little motorboat powering across the Mekong River to visit a fish farm. And this is what the inside of a fish farm looks like, or at least the fish farm manager's dwelling. At the back, you see the bedroom. To the left, the kitchen. You can just about see a vat here, so that's where the television lives. And then there's bags and bags of fish feed. And the fish are where it's wet, under the floorboards. And when you go through this dwelling, you come to the gangplanks, amidst neatly laid out cages with fish of different sizes, different ages, destined for different markets and different consumer preferences. And when you go up closer to these fish, you think, ooh, there's a person on the gangplanks. That must mean feed. And they all swim up to you and like, oh, where's our feed? Where's our feed? So these are actually very, very healthy fish. At the end of my talk, I'll briefly touch on vaccination. You can imagine that vaccination of fish, when you're talking about this number, can be a bit of a challenge. So most fish look very happy, apart from Meet Nemo. Sadly, in the Disney Nemo story, most of the fish die and Nemo lives. In my story, most of the fish live, Nemo does not. Nemo looks a bit goggle-eyed, and if you take a close-up of Nemo, that's what that looks like. It's known as Popeye in the aquaculture literature. The eyes of the fish are bulging. Exophthalmosis would be the technical term. There's opacity of the cornea. It's all blurry. You can't see very well. So we euthanized Nemo and checked whether he had Group B strep. In fact, first we put Nemo in a bucket. And you might think, why does a veterinarian who should be concerned about animal welfare not put enough water in the bucket? How hard can it be? Well, actually, there is enough water in the bucket. The reason the fish are swimming on their side is not because there isn't enough water in the bucket. These fish, like the gentleman in Singapore that lost his hearing, have meningitis. So their brains are not working well, so they don't know which way is up and which way is down and which way to swim. And that's why it looks like there's not enough water in the bucket for them to swim normally. So we euthanized the fish and put their tissue on culture plates and grew Group B strep. In fact, we grew the very same Group B strep that had been killing people in Singapore. So not only was there a link with the fish that was sold for consumption, which could have been contaminated after harvest, we know for a fact that fish actually get sick with that same Group B strep. And it's not just farmed fishes, lest you think it's all aquaculture's doing. No, it's not. It affects all sorts of fishes, from the very large ones to the teeny tiny ones. The one on the left is a giant grouper that was found in Queensland, and that's Rachel Bowater's team doing a post-mortem on it, and they discovered that this fish died of Group B strep. The one on the right are what we call pedicure fish or doctor fish, and they too can have Group B strep. And we think about one of my earlier slides where I said that Group B strep in adults can cause skin and soft tissue infections. Do we really want these Group B strep infected fish nibbling on our feet? Maybe not. I've been told that in Australia it doesn't happen. In Singapore, on the other hand, it's very popular. So the question arises, did we give Group B strep to that grouper? Can these nibbly fish give Group B strep to us? Which way does transmission happen? And it's not just fishes, it's all sorts of classes of animals. So at the top left, you see a ray. In Queensland, it wasn't just the groupers, but also rays that were affected. In the middle, at the top, amphibians, a different class of animals yet again, frogs. Crocodile, a reptile, very different type of animal again. In fact, there's an outbreak of Group B strep in farmed crocodiles in Australia that's been described in the literature. And when we continue with animals associated with water, we can also find mammals that are affected. That's a grey seal in Scotland, a dolphin, and a sperm whale. I actually have a Group B strep from a sperm whale in my collection. Now, for sea mammals, we don't actually know for certain whether it was Group B strep that caused their death or whether we just found it when we did a post-mortem. In fact, for the dolphin, we know that it was what killed the animal. And this happened in Kuwait Bay, where there was a sewage spill. There's a sewage treatment plant where effluent was unintentionally released into the water, which caused a massive fish kill and also killed a dolphin. And we think that with the seals and the whales, it's not so much that Group B strep kills them, but as they are dying and starting to inhale water, with the water, things like Campylobacter and Group B strep can enter our bodies, and that's how we find them at post-mortem. So it's probably us giving it to them in that case. And then on land, the story continues. Reptiles can be affected. Occasionally dogs and cats are affected, and it's mostly spillover from people. And this is a picture on the left, a big picture of a camel being milked. Now, camels are incredibly important for food security in the Horn of Africa. With the drought and the difficulty in maintaining dairy cattle in Australia, some Australian farmers are switching to dairy camels. And this is actually a picture from a dairy camel in Australia. And that finally brings me to the official name of Group B strep, because it's not officially called Group B strep. And I never talked about it as Group B strep for the first 10 years that I worked on it. Group B strep's official name is Streptococcus agalactiae, and I've already talked about that with a few of the audience members. Agalactiae being Latin for no milk. And that's because of what it does to the udder of the humble dairy cow. It infects the mammary gland and reduces the milk quantity and the milk quality. A cow infected with Strep agalactiae produces no milk, hence the name. And this, by the way, is the most beautiful udder in all of Scotland, photographed at the Royal Highland Show. Now, we've talked about One Health. Both Professor Ellen and I have talked about One Health and how people and animals and environment are all related and we're all in together. With Group B strep, it falls apart as soon as we start talking about it. Because medics know this organism as Group B strep, and most medics that I've interacted with in my research don't realize it's the same thing as Strep agalactiae. Conversely, most vets know it as Strep agalactiae, and very few of them know that that's the same thing as Group B strep. So, One Health, I'm all for it, but I have to keep explaining. So how do we untangle this mess of who gave it to whom, and when does it not happen, and when does it happen? That requires more than just detecting the organism. We know it occurs in people, we know it occurs in fishes, we know it occurs in cattle, but which ways do the arrows go? So to understand that, we've used DNA fingerprinting, and some of you will be very familiar with that. This is a drawing that my PhD student drew to illustrate it to non-professional audiences. So at the top you see the purple ball that is the Streptococcus. From that Streptococcus we extract the double helix of the DNA, and that double helix of the DNA is then sent through some type of sequencing device. In this case it's a MinION, a nanopure MinION, and that gives us the exact DNA sequence. And that's how we know the genetic code of Group B strep. And then we can ask questions like this: was it us? I mean, the giant groupers have Group B strep. We go snorkeling and scuba diving. We carry Group B strep in our urinary tract. You got to go, you got to go. So did we give it to the groupers? So the DNA fingerprinting is used to build family trees, and then we can position things in the family tree and see what's what. And most of you will be familiar with the concept of a family tree, with the great-grandparents and grandparents and the parents and aunts and uncles. And then there's the next generation with sisters and brothers and cousins, and then come the children and the nephews and the nieces and the grandchildren, all the rest of it. You can do the same for bacteria. So this is a family tree for Group B strep, and it's hideous. This is where the PowerPoint police are usually brought in to say, you can't do this in front of an audience. And one of the things that's really difficult with that particular tree is that it's so widespread. So one solution to it being so wide that it doesn't fit on the slide is to actually circularize it and make it a bit more tractable. So that's what I've done on the next slide. That's a circularized tree. And then actually in some families, let's say royal families, for example, there's sometimes a bit of sort of let's keep it in the family, the wealth, the countries, a bit of intermarrying. So it's not really a tree, it becomes a bit of a network. And that's what you see on the right, because bacteria do that too. They're not strictly tree-like. They actually exchange DNA and sometimes intermingle a little bit. And on that tree, you can then start to position the bacteria from the different host species. So here, for example, you see the baby next to number 17, and that is the major neonatal invasive clone. And that's one of the clones that is being targeted with the vaccine development. That type of Group B strep is almost never found in animals. I know of 2 exceptions, and I alone have typed thousands of Group B strep isolates from animals. And I like to think I know the literature reasonably well. There's virtually no evidence of that type occurring in animals. CC67 is a type that occurs in cattle, and time allowing I will get back to that. And then at the bottom you see an example of a type associated with fishes. So quite separate on that family tree. This is my preferred presentation of a family tree. This is officially an e-burst diagram, although when I talk to students I often call it a dandelion diagram. And I don't know how well that works in Australia, because they don't know whether you have dandelions. But you have the stem and then the little plumes. Yeah, people know a dandelion. Yeah. Can you see them all float away? Okay. So, dandelion diagram. And you can put the numbers on it again so that you know what is what. So, again, you can see number 17 associated with the baby. The cow now sits here in this presentation, so you can manipulate the impression that these diagrams leave a little bit with how you present them. And fascinatingly, the fish are in 3 different places. Top right, number 7, bottom right, number 283, and bottom left. And that's the one that we found in Singapore and in Vietnam. Now, when I look at a tree like that, knowing that almost every type can be found in people, and only those 3 types have been found in fishes, I try to read the tree leaves and ask myself, how come? How come that 3 so distinctly related, or distinct and distantly related types of Group B strep all infect fishes? And I'll give a more technical talk about that next month at Westmead, because my theory is that this is a case of a jumping gene. There's a particular gene that confers a survival advantage to Group B strep in fishes, makes the bacteria better at surviving in fishes, and that this gene has sort of skipped the branches of the family tree and jumped from one branch to another. We don't know where it started, we don't know whether it even came from a different bacterial species, but the 3 types associated with fish are the only Group B strep that have this particular jumping gene. There's a parallel in cattle, where we know that the jumping gene in bovine Group B strep is a gene that encodes the lactose, or it's an operon that encodes lactose use. Lactose is milk sugar. Where does Group B strep live in a cow? In the mammary gland. So all you need to be happy in a mammary gland as a Group B strep is the ability to use milk sugar, and that's the jumping gene in bovine Group B strep. So the last bit of the talk is then looking at journeys of various types. You can use family trees to build migration maps. And if you just Google family tree and migration, you get all sorts of maps like this, where you can trace your ancestry over shorter or longer periods. And this one goes all the way back to the proverbial Eve in Africa. And we can do similar things with bacterial family trees. So the first journey I want to share with you is a bit of an Australian journal. This work shown at the bottom here was co-authored by Nouri Ben-Zakour from the Westmead Medical Research Institute, and they looked at the genome of Group B strep in fish belonging to type 261, the type that was at the bottom left in the previous graph. And we think back to my slide with the FAO fish consumption curve that went up like that. The point where fish production in aquaculture really started to increase is around the 1970s, 1980s. And tilapia are a major species contributing to that growth. And tilapia originate from Africa, were first sort of commercialized in the Middle East. So Israel very kindly helped other countries and continents to establish their aquaculture industry by exporting tilapia. They exported tilapia and Group B strep to China. They exported tilapia and Group B strep to the US. They exported tilapia and Group B strep to Ghana. And they exported tilapia and Group B strep to Australia. And from the tilapia, it escaped into the rays. And from the rays, it escaped into the groupers. And this type is a uniquely fish-associated type. The groupers definitely got it from the rays and from the tilapias, and not from people, because this type uniquely has a smaller genome than normal Group B strep, and it cannot grow at our body temperature. So there's no record anywhere in the world that I'm aware of a mammal being infected with this type of Group B strep. So we can safely continue to snorkel and scuba dive with the groupers, at least from that perspective. Here's another one, and that's the dangerous one. This is the type 283 that killed people in Singapore. Until very recently, we thought that that type was uniquely restricted to Southeast Asia. And we've just published a paper about that, how for the past 2 decades, actually, this thing, unbeknownst to the medical world, was causing bloodstream infections in people in Thailand and other Southeast Asian countries. And it's only because of the outbreak in Singapore, where it popped up in a country that was sufficiently well organized and sufficiently well resourced to figure out what caused this outbreak, that we recognized this particular type of Group B strep as a cause of human bloodstream infection. Unfortunately, that type has now been exported from Southeast Asia to Brazil. And Brazilian researchers that published that finding say that one of the farmers that they worked with told them that they imported fish from Singapore. We've worked with the Singaporean authorities to try and trace that particular export record to verify that finding. We cannot verify that the fish came from Singapore. Singapore is an importing more than an exporting nation, so maybe a ship went through the port in Singapore, but chances are it came from somewhere in Southeast Asia rather than originally from Singapore. But the cat is out of the bag. The ST283 is now also in South America. And in South America, it's increasingly popular to invite people to come to fish farms and angle for their own fish. So they can catch their own fish and throw it on a barbie right then and there and enjoy that with their family. You don't want ST283 in that picture. Here's a different journey, the migration of language. So the countries shown in red here are united by a particular thing. And hint, it's not cricket. The North Americans don't understand cricket. All you need to do is read Bill Bryson and you'll know what they think of cricket. These are countries where more than 50% of the population speaks English. And it's not entirely surprising, maybe, because they were former British colonies. But with their colonists and their language, it seems that the Brits brought their cattle. And with their cattle, it seems that they've brought their Group B strep. And in all these countries, the type of Group B strep you find in dairy cattle is type 67. And when you read the scientific literature, you will read that bovine Group B strep is different from human Group B strep because this type 67 occurs worldwide in cattle and it never occurs in people. Well, again, that's the truth. I can't even say nothing but the truth. It's definitely not the whole truth. One thing, and I was going to say that especially if there were students present, but I think you're all sort of mature students, is that scientific literature is very biased. It's a culture in its own right. If you're white, male, and middle-aged, and from the right country, then your work gets cited. Speaking as someone who is white, but who wasn't always middle-aged, and who was never male, I can say that work from Northern Europe is definitely not cited by people in the UK and the US the same way they would cite work from their own countries. So the view that you get from the literature can be a bit biased at times. And if you ask the Viking what was going on, they tell you a very different story. We've done extensive typing of Group B strep from cows and people in Denmark, Finland, Sweden, and Norway. The most common types there are type 1 and type 23, which are shared by us and cows. And there is no host species barrier. There is no distinction. So which Group B strep you have and whether there's a link between adults and animals depends very much on which country, which culture, maybe even which language group you're in. Now there's another journey that comes into this story, and that's the journey of nutrients. To feed the world, we need to grow plants and we need to grow animals, but these animals and these plants also need to be fed. They also need nutrients. And these nutrients have to come from somewhere. If we just catch fish to feed other fish, that's not necessarily a very efficient use of nutrients. But waste, whether it's waste from pigs, from ducks, from chickens, or from humans, is actually a very rich source of nutrients. And I don't know whether anybody remembers the film Slumdog Millionaire and the toilet scene in Slumdog Millionaire. But this is sort of Slumdog Millionaire over a fish pond. And in Asia, that's actually how fish are fed. And in terms of recycling nutrients, that is a brilliant idea. That is recycling at its best. Unfortunately, some bacteria think so too, and it probably explains why the Group B strep strains that are shared between people and fish, until recently, were only found in Asia, because Asia is the only part of the world where this method of feeding fish is used. In the rest of the world, it was only the fish-specific Group B strep. In Asia, types of Group B strep emerged that are shared between people and fishes. And I think that this has something to do with it. And that was just a pond, which is more or less contained. The same story applies on the Mekong River. I was there long enough that I needed to use the facilities at some point, and that little red outhouse that you see there, that's the facilities. So everything goes straight into the water, and it's not specifically used to feed the fish, but the fish are in that water too. The fish are under that grey building, and the outhouse is attached to it. And that's not contained in a pond, that's flowing gently downstream. So there is Group B strep coming out of us, going into the water, potentially infecting fish. Fascinating ecology, if you're into that sort of thing. And that brings me to the last topic of the talk, challenges and solutions. And there's some major challenges. In this country, I don't have to explain the challenges that climate change and environmental change bring. Much as politicians don't like to admit to it, I think it's undeniable that global warming and environmental change has a lot to do with the current fire season and the drought. I won't talk about that in Australia, though. I'll take you back to Southeast Asia, where population growth is immense. And as we said, all these people need to eat. All these people also need water. So what you see here, hopefully you can see it. I don't know what I can point out now. There's a blue, wrong one. There's a blue squiggly line running sort of top to bottom through this graph. That's the Mekong River. And all the dots along that line are dams that the Chinese have built, are building, or are planning to build to extract water for their population. Which means that there's not going to be a whole lot of water left for Southeast Asia downstream of China. So water levels are decreasing, pollution levels are increasing, so there's less water coming down the river, and at the same time, as seawater levels are rising, particularly in the Mekong Delta, there's more salt water coming up the river. So you can imagine what happens, the water becomes saltier and saltier. Now, tilapia are a very attractive aquaculture species in that sort of environment because they put up with a little bit of abuse. They can withstand some pollution in the water. They can also withstand some salinity in the water. So even under these conditions, you could still farm tilapia. They will be a bit challenged and immunocompromised, but at least they can still live and grow. Unfortunately, Group B strep can also live and grow in fresh water and in salt water. So the Group B strep will continue to be a threat and increasingly be a threat as environmental change reduces the water flow, increases the salinity, and compromises the immune system of the fish, which we still need to feed that growing population. Challenges on the disease side, whether it's human disease, cattle disease, or fish disease, are the need for vaccines, the need for better diagnostics, and the need for treatment. You've all gone very quiet, and you're all starting to look a bit dejected. So I think it's time that I start talking about solutions. This is one solution, a local solution, a Sydney solution. One of the things we can do to address issues of food security and resource and nutrient scarcity is be cleverer about how we produce our food. So having hydroponic horticulture, growing plants in greenhouses, and then using the heat of that greenhouse to also create the environment where we can farm fish is something that increasingly happens. These photos are from Green Camel, which is in Cobbitty, right down the road from our Camden campus. And there are similar efforts going on in the US, in Singapore, in the Netherlands, co-culture of herbs and vegetables and fish. Very small spatial footprint, also a very small carbon footprint. So these are the sorts of solutions that we need to continue feeding the world. Solutions in terms of vaccines. So the vaccine I talked about that was presented at the meeting in South Africa is currently in a phase 1 and 2 trial, which means that they're administering it to people, to healthy volunteers, to check its safety and to check that it induces antibodies. And once it's been shown to be safe and to induce antibodies, then the trial will move to the next phase where they'll be administering it to pregnant women to see whether it actually provides protection to babies. That's a fantastic success story. For some people, that is a career that spans decades to finally get to that point. It's not the be-all and end-all. This meeting was held in Africa, and in the final session of the conference, there was a discussion forum about uptake of this vaccine. And the idea of the developers and funders of the vaccine is that we'll demonstrate how good it is, and then people will want it and use it. That's not necessarily how household economics in sub-Saharan Africa work. So there are still hurdles to be overcome, and I think it's incredibly important that as medical and biological scientists, we collaborate with social scientists to not only develop the tools, but to also understand the drivers and inhibitors for uptake of those tools in society. For the fish, well, what we're trying to do, remember that jumping gene that I talked about, the little bit that's essential for causing disease in fish? Initially, we came to that conclusion just on the basis of comparison of genomes. Then we said, well, we better check our findings, so we made a mutant bug where we knocked out the bit that is essential to fish. And lo and behold, fish with the intact bug die. Fish with the mutant bug, where you've taken out the jumping gene, they live. So this bit matters. Sadly, we don't know what it does, and we don't know how to bottle it and administer it to fish. So we're still working on a fish vaccine. There are vaccines on the market, but they only protect against one type or the other, not both. And farmers can't afford to spend their money on the wrong vaccine, so they don't vaccinate at the moment. So that's a work in progress and something that I personally hope to progress here in my new position in Sydney. And then finally, we also need to know whether people, cows, fishes have Group B strep. And ideally, we know this at the point of care. So ideally, I walk down those gangplanks on the fish farm, take a fish out of the water, get a bit of tissue, put it on the test, and know right then and there, this is the vaccine you need to order. Actually, when I was on the fish farm, the fish farmer was on his tablet ordering vaccine from a pharmaceutical company, and he complained that the vaccine doesn't always work. And based on the testing we did on his fish, I now know that he was ordering the wrong vaccine. So ideally, we have this pond side, or in human healthcare, bedside for women in labor. The current testing that's done in the US is done a few weeks before expected labor and birth. In the UK, they don't like that idea, so that testing is not done. Australia has a mix of the two systems whereby people can choose whether or not they want to be tested. But there are issues around the timing of that testing. An ideal time, if there ever is an ideal time to detect pathogens, will be during birth. So we are working with the NICE, the chair of the Group B Strep Committee of NICE in the UK, to develop a test that can be used at the point of care. And this is very clever engineering. This photo was taken by my colleague Julien Rebou, who's a bioengineer in Glasgow. And this is essentially a lab on a chip, only the chip is made from paper. It's a lamp assay, for those of you interested in the technicalities. There's a lot of clever microfluidics with printing and waxing and folding and all the rest of it. But this stuff is so cheap that you can have a whole pile on your desk. You can put it in your pocket, you can take it to a fish farm, you can use it to test the fish, or they take it to Uganda to test people for malaria. And rather than creating a biohazard because you're growing this thing on a farm, you can, after you've done the test, just burn it, and it's safe, and you know exactly what you're dealing with. So that's a sort of solution that is starting to come within reach in terms of diagnostics. And then the last thing we need is treatment. in this presentation, I haven't talked about antimicrobial resistance very much. And until recently, antimicrobial resistance wasn't really an issue for Group B strep. Unfortunately, that is now also starting to change. And in Japan and China in particular, reports are coming out of penicillin-resistant Group B strep in people, in cattle, and in fishes. So antimicrobial resistance is becoming a bit of a concern. What's probably a bigger concern, and the reason that in the UK they're reluctant to offer blanket screening and treatment, is that when you treat someone with antibiotics, that impacts not just on the bug that you're trying to treat, it impacts on everything. Everything on my skin, everything in my lungs, everything in my guts, everything is affected if I'm being given systemic treatment. And we're increasingly learning that changes to the microbiome, all the creepy crawlies that live in your gut, so to speak, and in your lungs and your skin and everywhere else, can result in disease in later life. Things like eczema, asthma, even obesity in children are potentially linked to changes in the microbiome that could be brought about by treatment to prevent Group B strep disease. So you're solving one problem, potentially creating the other problem. So what we're trying to do is develop a precision treatment that only targets Group B strep. And the lady you see here is Ploy Krawanyong, who's a PhD student that just finished her PhD in Glasgow. And the funny bit on the left is the insides of a wax moth larvae. It's essentially a caterpillar that we managed to infect with Group B strep, and she used that model to show that the product that she's developing kills the Group B strep, but not anything else. So we're trying to work that up as a very specific targeted treatment for Group B strep that doesn't have negative consequences for the rest of the bacteria in the body. So it's sort of precision medicine for bacteria. I think my time has been up for a long time, so I'll draw this to a close, and I'll leave you with a few take-home messages. If you or your partner or a friend or a colleague are pregnant and want to talk about Group B strep, speak to your healthcare professional. If you travel to Southeast Asia, and I do recommend it, enjoy the beauty of the countries, the friendliness of the people, and their delicious food, but be careful with what you eat. And even when you ask, be careful in what you ask, because one of the things that we've learned in our research in Singapore is that when we talk about fish that's cooked, we mean fish that is heat treated so that the bacteria are killed. In a lot of cultures there, and languages there, the word cooked means something slightly different. It means prepared. So a raw fish porridge is a prepared dish. It's not just a raw fish that you buy at the market stall. It's a prepared dish. And the word cooked and prepared are treated as synonyms. So where I'm talking about, has it been heated and have the bugs been killed? They just say, mean it's been cut up and presented nicely with some very nice fresh herbs. So be careful and bring your dictionary or your friendly anthropologist if you're really worried. And finally, we're all in it together. Human population growth drives migration, water extraction, pollution, and the need for more food. And they may all conspire to give Group B strep more opportunities to infect people and animal. And there's some fantastic science going on to try and overcome these problems. And Sydney is part of that. And I very much hope to become part of that here in Sydney too. And with that, if there's time, I'll thank my many collaborators, far and wide. I haven't covered all 6 continents because I skipped the dairy cows in Colombia. But I've had funding from industry bodies dealing with cows and with fish. We have had funding from universities supporting PhD students. We have international funding from FEO and the Global Challenges Research Fund and the Learned Societies in the UK. And that's specifically for the work in Southeast Asia. And the Medical Research Council has funded some of the work on diagnostic test development. I hope we can build on those achievements and take them further here. And with that, I'll shut up and I'll be happy to take questions here or by email. Thank you very much.
Thank you, Ruth, for a really entertaining and thoroughly enjoyable talk. I'll thank you formally in a moment, but I do think we have time for some
Hi, the wonderful talk. I'm just wondering, you were mentioning that the checking of the B strep is best at birth. Wouldn't it be too late if it's already infected, both the mother and baby? Wouldn't you want to do it a bit earlier?
Yeah. So again, I have to be careful in what I say here because I'm not a medical doctor. But the idea is that labor usually takes a while. So if we can detect bacteria at the onset of labor, there's enough time left to intervene. If we detect the bacteria after birth, we could still monitor a baby closely and intervene as soon as there's a suspicion that the baby is not doing as well as it should do. But I take your point. Ideally, the diagnosis around birth is made a few hours before birth at the onset of labor. And I don't know enough about the situation in Australia, but I know in the UK there's a growing interest in demedicalization of birth and giving people the opportunity to give birth not in a hospital, but either at home or in a birthing clinic under the supervision of a midwife. And the idea is that this will be a tool that we could put in the hands of midwives to allow labor and birth to take place in a more home-like setting while still mitigating the risk of Group B strep disease.
Are there any more questions?
This one here.
Hi, I just wanted to ask about the solutions at the end. In the research for precision treatment, I think you said you were had infected a caterpillar. Was that correct?
Yeah.
I was just wondering personally, why was that a caterpillar chosen as an animal model versus any other types?
Yep, and that's a very good question. So why did I use a caterpillar model rather than another animal model? And that's because caterpillars, I think, are not considered to be sentient beings. Most of the animals that have been used for research are creatures like mice and rats and guinea pigs, who are social creatures with a certain level of awareness. So there's a strong drive to reduce the use of such animals in experimentation. And caterpillars are considered to have a much lower cognitive status, so there's not the same level of concern about using them in research. So we used the caterpillar as a first stage of screening, and they're much better than, say, just growing things in a petri dish, because caterpillars are actually living creatures that have an immune system and to some extent reflect the severity of disease caused by the different types. So we were able to show that the type that kills babies and the type that kills adults are also more likely to kill the caterpillars than any other type. So to some extent it reflects what happens in other species, but without the sort of damage or animal suffering that might be associated with testing on other species. So that's why we've used the caterpillars.
Just talk. A silly question. Do you eat sashimi?
Pardon?
Do you eat sashimi? Japanese raw
well,
food.
I'm
I'm
Dutch.
interested.
I prefer raw herring. So yes, I eat raw
But
fish.
you would eat raw fish.
I eat raw fish. Yes, I do. Maybe less so now in Southeast Asia, and I'd be a bit careful in which fish I eat. As far as I know, there's never been any Group B strep in salted herring. So that's the one I eat. But I think you actually raise a very serious point, because when we started talking about this at a meeting of Southeast Asian experts, you're like, oh yeah, well, who eats raw fish? I mean, that's silly. That's taking a risk with your life. Well, I think actually a lot of people eat raw fish. And yes, I'll put my hand up. I'm one of them.
Very good. Ladies and gentlemen, I'd like you to join me in thanking Professor Ruth Zadoks for a fascinating talk. I really like to dwell on that point she said last. We are all in this together. it is important to, I guess, and Ruth has illustrated this wonderfully well, the role that veterinarians do play in the health of not only animal populations but that of people as well. And Ruth has dedicated her research career to just that point. And there are many more veterinarians around the globe who are doing just that. So Ruth, fascinating work. We're delighted to have you here at the University of Sydney. And thank you very much for giving a very thorough and entertaining talk about Group B Strep. Thank you. So thank you everyone, and safe travels, and please stay safe, particularly tomorrow.